Hello everyone, in today's video we will be going to discuss paper 1 of AS level biology 9700. This is of year 2021 and this is the winter paper or October-November paper. So this is October-November 2021. Let's start. MCQ number 1. A scale bar on an electron micrograph is 2 centimeter long and represents an actual length of 1 micrometer. So they say that the scale bar on the electron micrograph, which is a photo taken from the electron microscope, the length of the scale bar is 2 centimeters. And the actual length is 1 micrometer. So the length of the scale bar is the image length, that is 2 centimeter, and actual length is 1 micrometer. So what is the magnification of the electron micrograph? Magnification is equal to image length divided by actual length. Always remember that the length of the scale bar that we measure is the image length, and the actual length is usually written near the scale bar, or the examiner will tell you the actual length. Let's calculate the magnification. Image length over here is in centimeters, so we need to convert the image length into micrometers. So how do we convert 2 centimeter into micrometer? What we can do, we can first multiply it by 10 to convert it into millimeter, and then we can further multiply it by 1000 to convert it into micrometers. Then we will divide by the actual length, that is 1 micrometers. So the answer will be into 20,000. Magnification will be into 20,000. The answer is C. Okay, let's move on to the second MCQ. This is a very good MCQ. The eyepiece of a microscope is fitted with an eyepiece graticule. So what is meant by this statement? The eyepiece of a microscope is basically the part of the microscope through which we view the specimen. It is the topmost cylindrical part of the microscope through which we can see an image. So in the eyepiece, a scale is fitted which is known as eyepiece graticule scale. You all know about this scale and you might have used it various times in the lab. Okay, what is the next statement? And a stage micrometer scale is placed on the microscope. What is meant by this statement? That a stage micrometer scale is placed on the microscope. This means that the slide right now, which is on the stage of the microscope, consists of stage micrometer scale. It means there is no specimen right now. We can only put one slide at a time on the stage on the microscope, on the stage of the microscope, we can only place one slide at a time, right? So right now, the slide containing the stage micrometer scale is placed and the specimen slide is not placed right now. Which statement about the stage micrometer scale are correct? Number 1, the scale can be used to measure the actual length of the cells directly. This is wrong. Let me tell you why. As I just mentioned, that we can place one slide on the stage of the microscope at any one time. So we can either place the slide of the specimen. For example, I placed a slide of plant root. So in that case, I will be able to see the cells of the plant root, right? So this is the slide of the specimen. If I want to view the stage micrometer scale, I have to remove the slide of the specimen, that was the plant root, and I have to place the stage micrometer scale slide to view the stage micrometer scale. Basically, I cannot view the stage micrometer scale and the specimen together. I hope you understand this point. I'm repeating it again. Either I can place the slide of the specimen, for example, the plant root. For example, I have to measure the length or the width of the xylem vessel, right? So I cannot measure the width of the xylem vessel using the stage micrometer scale directly. Why? Because either I can put the slide of the plant root to view the xylem vessel, or I can place the stage micrometer scale. I cannot place the two slides at a time. So I can never use the stage micrometer scale to measure the width or length of the cells directly. What I can do is that I can place the slide of the specimen first, and then I can view the xylem vessel, and then I can measure the length of the xylem vessel or the width of the xylem vessel using the eyepiece graticule. But as you all know, that eyepiece graticule is not a true scale, so it must be calibrated. Okay, one more thing, that I can view the specimen together with the eyepiece graticule. Why? Because eyepiece graticule is a scale that is fitted into the eyepiece. We don't use a slide for it. This is fitted in the eyepiece, and I can still place the slide of a specimen on the stage. So we can view the specimen and the eyepiece graticule scale together, but we can never view the specimen, that is the plant root, and the stage micrometer scale together. So what we usually do is that we first place the slide of the specimen. We measure the length or the width of the cell using the eyepiece graticule. And since eyepiece graticule scale is not in the true units, we calibrate it using stage micrometer. How do we calibrate eyepiece and stage? What we do is that we remove the slide of the specimen, we place the slide of the stage micrometer scale, and then we place the eyepiece graticule scale and the stage micrometer scale together, because we can do that. The slide is of stage micrometer scale, and in the eyepiece there is eyepiece graticule. So we can match the two scales and calibrate them. I hope you are getting my point. In a separate video, I will teach you how to use these two scales together. How do we calibrate? I know many students have a problem in these types of questions. Many students have a problem in eyepiece graticule and stage micrometer scale understanding. So hopefully in the separate video, I will be going in detail, and I will show you various pictures and other illustrations to make this crystal clear for you. Okay, so the first statement is wrong, which says that the scale can be used to measure the actual length of the cells directly. No, stage micrometer scale cannot be used to measure the actual length of the cells directly. It can be used to measure the actual length of the cells indirectly. First, you have to measure the length of the cells using eyepiece graticule, then you have to calibrate the eyepiece graticule scale with stage micrometer, and then you have to find the actual length. This scale allows you to calibrate the eyepiece graticule. This is the true statement. Less of the scale is visible as the objective lens changes from into 10 to into 40. This is true. Obviously, when you change the objective lens of the light microscope from into 10 into 40, so magnification increases. And as you all know that the stage micrometer scale is on a slide that we place on the stage of the microscope. So when you magnify, the scale will also magnify, and less of the scale will be visible. So statement 3 is right. The answer is B, 2 and 3 only. Okay, let's move on to MCQ number 3. This is also a good MCQ. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. So what we have to do, we have to match the function with the appearance of some cell structures. Right, the functions were listed by the number. mRNA passes through to the ribosome. So through which structure mRNA passes to go to the ribosome? The answer is nuclear pore in the nuclear membrane. So in order to go to the ribosome, the messenger RNA must pass through the nuclear pore, which is found in the nuclear envelope. That's because mRNA are produced in the nucleus by the process of transcription. Okay, this was about 1. Organizes microtubules to produce the spindle fibers during cell division. This is done by centrioles. This is the function of centrioles. Synthesis of polypeptides is the function of the ribosome, as you all know. Okay, fine. Let's move on to the appearances. Membranes which surround an enclosed inner cavity. Okay. Non-membrane bound spherical structures, these are definitely ribosomes. If we talk about spherical structures which are non-membrane bound, so these are ribosomes. A double membrane interspersed with pores. This is nuclear envelope, which is a double membrane that is interspersed with nuclear pores. So this is nuclear envelope or nuclear membrane. Okay. Non-membrane bound cylindrical structures, these are definitely centrioles. Okay. Membrane bound sacs arranged as flattened sac. This could be Golgi apparatus, or this could be endoplasmic reticulum, right? Most probably this is endoplasmic reticulum because these are flattened sacs and they haven't mentioned that they are curved. If they were curved flattened sacs, we could say Golgi apparatus. Most likely this is endoplasmic reticulum, and V states that membranes which surround an enclosed inner cavity. This could be many organelles. This is not specific appearance. So let's match the appearances with the functions. Okay. So as we all know, synthesis of polypeptides occurs in ribosomes, that is W. So for 3, the option is W. Okay. And mRNA passes through to the ribosomes, that is the nuclear membrane, which is X. So 1 is X, and organizes microtubules to produce the spindle during cell division. This is centrioles. And so 2 is which one? Centriole was Y. So 2 is Y. The answer is D. Okay, let's move on to MCQ number 4. Which cell structures are required for the formation of lysosomes and the hydrolytic enzymes that they contain? So guys, for the formation of hydrolytic enzymes, which are proteins, we need rough endoplasmic reticulum as it contains ribosomes. For the formation of these proteins, obviously ATP are required, which come from mitochondria. And for the formation of lysosomes, you need Golgi body. So we need the 3 of these organelles to form hydrolytic enzymes and the lysosomes that contain these enzymes. So the answer will be B. Which cell structure contains ribosomal RNA? Always remember that the cell structures which contain ribosomes also contain ribosomal RNA. Ribosomal RNA is used to make what? Used to make ribosomes. So always remember that a cell structure which contains ribosomes contains ribosomal RNA. So always remember that chloroplast contains ribosomes. So it contains ribosomal RNA. Chloroplast contains 70s ribosomes. Mitochondria also contain 70s ribosomes. So they contain ribosomal RNA. What about the nucleus? Nucleus also contains ribosomes, but the ribosomes do not function in the nucleus. They come out of the nucleus and they function outside the nucleus, right? They are either attached to the nuclear membrane or the rough endoplasmic reticulum, or they are found in the cytoplasm. But you all should know that ribosomes are made in the nucleus, in the nucleolus of the nucleus. So nucleus also contains ribosomes or ribosomal RNA. Smooth endoplasmic reticulum does not contain ribosomes, and hence it does not contain ribosomal RNA. So the answer is B, 1, 2, and 3 only. The single-celled organism Theomargarita namibensis measures 700 micrometers in diameter and is enclosed in a cell wall. The genetic material of T. namibensis is located free in the cytoplasm, where it occurs as thousands of copies of circular DNA. Most of the cell is taken up by a large vacuole which stores essential chemicals for metabolism. Which statement is correct? It must be a eukaryote because a diameter of 700 micrometer is too large for a prokaryote. Basically, this is a wrong statement. I know that you all have studied that the diameter of the prokaryote must be less than 7 micrometers. It's mentioned in the books and in the marking scheme as well that the diameter of the prokaryote must be less than 7 micrometers, or it should be between 0.5 to 5 micrometers. But always remember that we can still invent those prokaryotes which could be larger. Obviously, the scientists are still inventing many new species. So up till now, the prokaryotes that have been invented have a diameter less than 7 micrometers. So we cannot say we might invent a new species of prokaryote which could have a diameter of more than 7 micrometers. So we cannot say straight away that if a cell is very large, for example 700 micrometer, it could not be a prokaryote. To identify a prokaryote, we must look at the features, we must look at the cell structure, right? So never ever make your conclusion on the basis of size. Look at the features of the cell, right? It must be a plant because it contains a vacuole. This is not true. Only plant cells, it's not necessary that only plant cells will contain a vacuole. Other cells can also have a vacuole. For example, the cells of the fungus, they also contain vacuoles. Even animal cells contain vacuoles. Those are temporary vacuoles, not the large central permanent vacuole, but other cells can also contain vacuole, right? So it's not necessary that if a cell contains a vacuole, it's a plant cell. It must be a plant because it is enclosed in a cell wall. It's not necessary that if a cell contains a cell wall or if a cell is enclosed in a cell wall, it must be a plant cell. It's not necessary. It must be a prokaryote because the DNA is circular and is located in the cytoplasm. This is a perfect statement. It must be a prokaryote because, as we just read in the above information, it says that the genetic material is located free in the cytoplasm. And it occurs as thousands of copies of circular DNA. So DNA is circular and it lies freely in the cytoplasm. This is the feature of a prokaryote. So it is definitely a prokaryote, right? So D is the best statement. MCQ number 7. A student carried out 4 tests on a sample of biological molecules. What conclusions made by the student are correct? Okay, when the student performed emergent test, it appeared cloudy. That means fats were present. This is true. As you all know, if we carry out an emergent test and if the emergent turns white or cloudy, it means fats are present. Okay, when we carried out Benedict's test, the color changed from blue to yellow. This means that reducing sugars are present. This does not signify that glucose was present. Any reducing sugar could be present. Benedict's test is for reducing sugars. Benedict's test is specific for reducing sugars and not for glucose. Always remember that. Whenever you get a positive Benedict's test, it does not mean that the solution contains glucose. It might contain other reducing sugars like fructose, like galactose, like maltose, like lactose. There are other reducing sugars as well. So it's not necessary that whenever the Benedict's test is positive, glucose is always present. It's not necessary. Protein was present. Okay, let's see. When we carried out biuret test, observation was purple. It means that the biuret reagent turned purple from blue. That means proteins are present. When we carried out iodine test, the color stays yellow, which means that starch is absent. Starch was not present. So statements 1, 3 and 4 are correct. Statement 2 is wrong. So the answer is C. Which general formula is correct for monosaccharides such as fructose? So as you all know, and I also mentioned in my lectures, that the general formula for monosaccharides is CH2O bracket N, where N is the number of carbon atoms. So the answer is B. Which statement can be used to describe the structure of cellulose? A polymer of glucose monomers, this is true, linked by beta-1,4 glycosidic bonds, this is true. If we talk about cellulose, cellulose is made up of beta-glucose molecules which are joined by beta-1,4 glycosidic bonds. It's a polysaccharide of hexose monomers, definitely. Glucose are hexoses, so this statement is true as well. Cellulose is unbranched, definitely. Cellulose is an unbranched macromolecule which is made up of beta-glucose monomers. This is also true. So the answer is A. Let's move on to MCQ number 10. Complete hydrolysis of polysaccharides requires all the glycosidic bonds between the monomers to be broken. Enzyme X only breaks alpha-1,4 glycosidic bonds. Which row shows how completely enzyme X can hydrolyze molecules of glycogen and amylose? So in this MCQ, they are saying that the complete hydrolysis means that all the bonds between the monomers are broken, right? And if we talk about hydrolysis of polysaccharides, so all the glycosidic bonds between the monomers must be broken. That is complete hydrolysis. And then they're saying that there is an enzyme X which only breaks down alpha-1,4 glycosidic bonds. So which row shows how completely enzyme X can hydrolyze molecules of glycogen and amylose. So guys, as you all know that glycogen contains which kinds of bonds? Glycogen contains alpha 1,4 and alpha 1,6 glycosidic bonds, whereas amylose contains only alpha 1,4. So this enzyme can completely break down amylose because this enzyme can break down alpha 1,4 glycosidic bonds. However, this enzyme will be able to break down glycogen but not completely. This enzyme X will break down glycogen partially because this enzyme will break down all the alpha 1,4 glycosidic bonds, but this enzyme will not be able to will not be able to break the alpha 1,6 glycosidic bonds. So for amylose, there will be most hydrolysis, and for glycogen, there will be some hydrolysis. We cannot say no hydrolysis. Negative means no hydrolysis. Enzyme X will hydrolyze glycogen because it will break down alpha 1,4 glycosidic bonds, but enzyme X will not be able to break alpha 1,6 glycosidic bonds. So the answer is A. Let's move on to the MCQ number 11. Which molecules contain at least 2 double bonds? So they are saying double bonds. They did not say carbon-carbon double bond. It means they are talking about any double bond, right? If we talk about saturated fatty acid, so saturated fatty acid does not contain any carbon-carbon double bond, but it contains one carbon-oxygen double bond, as you all know. If we talk about saturated fatty acid, so saturated fatty acids do not contain any carbon-carbon double bond, but they do contain one carbon-oxygen double bond, right? Okay. If we talk about collagen and hemoglobin, so they contain amino acids and they contain many amino acids. Both are proteins and each protein has a carboxylic acid group. Each amino acid has a carboxylic acid group. You all know that. So each amino acid contains a double bond, yes or no? Right? If we talk about proteins, so proteins are made up of many amino acids and each amino acid contains a carboxylic acid group with CO group in it. So if we talk about hemoglobin and collagen, they have many double bonds. But if we talk about saturated fatty acids, they only have one double bond. We have to select an option which contains at least 2 double bonds. So saturated fatty acids will not be the answer because they only contain one double bond. However, hemoglobin and collagen, they are proteins, they are made up of many amino acids joined together. So they contain at least 2 double bonds, or we can say more than 2 double bonds. So the answer will be C. So always be careful. In this MCQ, they stated double bonds only, not carbon-carbon double bond. Any double bond. They're talking about any double bond. Okay, MCQ number 12. The diagram shows an amino acid. Which group is changed to produce different amino acids? Always remember that the basic structure of amino acid is that each amino acid contains a constant hydrogen atom, a constant carboxylic acid group, a constant amine group. The thing which is different is the R group. In this case, R group is CH3. All the other groups are constant. So which group is changed to produce different amino acids? The CH3 group is changed. So in this amino acid, we have the CH3 group. In other amino acids, we will have different groups rather than CH3. So R group is changed to produce different amino acids. Which row is correct about the structure of proteins? Okay, if we talk about the primary structure, let's look at the statements and we will take those statements which are right and we will cross those statements which are false. Okay, determined by the sequence of DNA nucleotides, this is true. Why? Primary structure of proteins is basically the sequence of amino acids. If we talk about the primary structure of proteins, it is the sequence of amino acids in a polypeptide, or we can say how amino acids, in which sequence amino acids are attached. So as you all know that the DNA nucleotide sequence in a gene determine the sequence of amino acids in a polypeptide or protein. So primary structure is always determined by the sequence of DNA nucleotides of a gene. So this is the true statement. The number of amino acids in each polypeptide chain, no. The primary structure does not depend on the number. It depends on the sequence. Two proteins may have same number of amino acids, but those proteins may have a different primary structure, because the primary structure is the sequence of amino acids in a polypeptide formed by 20 different amino acids linked in a specific order. This can be true for those proteins which contain all the possible amino acids, that is the 20 different amino acids. This is fine because they have mentioned that these amino acids are linked in a specific order. The sequence of amino acids in each polypeptide chain, this is also true for the primary structure. So the options could be A, C, and D. Okay, let's look at the secondary. Occurs by hydrogen bonding between NH and CO groups of amino acids. This is very true. As you all know, that secondary structure, the secondary structure could be alpha helix, could be beta pleated sheets, and these are formed due to hydrogen bonding between CO groups and NH groups of amino acids. In a single polypeptide, it is either an alpha helix or a beta pleated sheet. In a single polypeptide, it is either an alpha helix or a beta pleated sheet. This is true. Not exactly true. Both can occur in a single polypeptide. In a single polypeptide, both alpha helix and beta pleated sheets can occur. So this is wrong. An alpha helix is formed by hydrogen bonds between side chains of amino acids. This is wrong again. Side chains of amino acids. In secondary structure, hydrogen bonds are not formed between the side chains of amino acids. Hydrogen bonds are formed between CO and NH groups. If we are talking about tertiary structure, then definitely hydrogen bonds are formed between the side chains of amino acids. Okay, a beta pleated sheet is the result of a folded polypeptide forming hydrogen bonds between adjacent strands. This is wrong. Not adjacent strands, it is the same strand, not the adjacent. Adjacent means that they are talking about hydrogen bonding between 2 or 3 neighboring strands or adjacent strands. When we talk about secondary structure always forms within the strand, not between the strand. I hope you get this point. If we are talking about secondary structure, we are talking about one polypeptide chain, not adjacent strands. We are talking about a single strand. Okay, this means that option is A, because the remaining options include the statements of secondary structure which are wrong. Let's look at the tertiary structure statement. Disulfide ionic hydrogen bonds and hydrophobic interactions are all involved. This is true for tertiary structure. Quaternary structure is always formed by the association of 2 or more polypeptides. This is true as well. Quaternary structure is formed when there are association of 2 or more polypeptide chains. So the answer is A. MCQ number 14. Which graph correctly shows possible changes in the energy levels as a chemical reaction progresses with or without enzyme? So you all should know that the enzyme, what enzyme does, it lowers down the activation energy. However, there is no change in the energy level of the substrate and the energy level of the product. The initial energy level of the substrate and the final energy level of the product should be same. There should be no change in the enthalpy. Enthalpy change will stay constant. Only the activation energy will fall. So the dotted line is without enzyme and the firm line is with enzyme. Okay. So the two graphs should meet at the start and at the end. However, when you are using an enzyme, activation energy should fall. This means the answer is C. Right? So with enzyme, you will see a fall in the activation energy. How do we find the activation energy? Let me tell you. If you want to check on the activation energy, extend the reactant level and draw an arrow from the reactant level to the maximum point. So you can see that for the dotted line, the activation energy is higher, and for the firm line, the activation energy is lower. And this is true that with enzyme, activation energy decreases. However, the graphs meet at the energy level of the substrate and the energy level of the product. So the answer is C. Let's move on to the MCQ number 15. The graph shows the relationship between the concentration of substrate and the rate of an enzyme catalyzed reaction. We have rate of enzyme catalyzed reaction on y-axis and we have concentration of substrate on the x-axis. Which row shows how Km and Vmax for this enzyme would be affected if the same reaction was carried out in the presence of a competitive inhibitor? Guys, always remember that if a competitive inhibitor is used, so what are the changes in the graph? The graph somewhat becomes like this. So what we observe that the Vmax stays the same. Vmax is the maximum initial rate of reaction. It stays the same with competitive inhibitor. What happens to the Km, however? Km increases. Km is defined as the substrate concentration at half Vmax. So if we take the half value of Vmax, which is half Vmax, so the substrate concentration on half Vmax is the Km. So for the black graph, Km is lower, and for the blue graph, Km is higher. So always remember that the competitive inhibitor have no effect on the Vmax. So Vmax remains the same, however, Km increases. So the answer is B. Liver cells, MCQ number 16. Liver cells contain vesicles that have proteins in their membrane, which are specific for the transport of glucose. When these cells need to take up glucose, the vesicles fuse with the cell surface membrane. How does the uptake of glucose occur? Guys, as you all know, glucose is a polar molecule, right? So glucose will be taken up by facilitated diffusion through transport proteins. They have also mentioned that liver cells contain vesicles. What they are saying in the MCQ, that liver cell contains vesicles which contain proteins that are specific for the transport of glucose, and obviously these vesicles contain transport proteins for glucose which are involved in facilitated diffusion. Proteins are not involved in exocytosis. They are involved in diffusion, but which type? Facilitated diffusion. And transport proteins are not involved in endocytosis as well. So the answer will be D, facilitated diffusion. Okay, let's move on to MCQ number 17. This is a very good MCQ. A student set up an experiment to investigate diffusion. A block of agar 1 centimeter by 1 centimeter by 1 centimeter was stained uniformly with a water-soluble blue dye. The block of the agar was put into a test tube containing 10 centimeter cube of distilled water at 20 degree Celsius. The intensity of the blue color of the water after 5 minutes was measured. So let me explain you this experiment. What the student is doing, student is investigating the diffusion, and student took a block of agar jelly which was 1 centimeter by 1 centimeter by 1 centimeter, which means that the block has a length of 1 centimeter, width of 1 centimeter, and height of 1 centimeter. And this block was stained with a water soluble blue dye, right? So the block turns blue, but this dye is water soluble. If we put the block in the water, the dye will dissolve in water and the block will turn colorless, right? The block of the agar was put into a test tube containing 10 centimeter cube of distilled water at 20 degrees Celsius. This means that because the blue dye is water soluble, so it will leave the agar block and it will dissolve in water. So what the student is observing is the intensity of the blue color of the water after 5 minutes. So the student placed the block which was stained with a water soluble blue dye in water, and the student measured the intensity of the blue color in the water after 5 minutes. 4 other experiments A, B, C and D were then carried out using different numbers of agar blocks, different sizes of the agar blocks and different temperatures. All other variables were standardized. Which experiment would give a lighter blue color in water after 5 minutes compared to the first experiment? So what is the examiner asking in this question? That which experiment would give a lighter blue color? This means that less blue dye escapes the blocks and goes in water. So in which case will that happen? We need to compare these options with the original block, right? So which experiment would give a lighter blue color in water after 5 minutes compared to the first experiment? Okay, so we have to compare it to the first experiment. In the first experiment, the temperature was 20 degrees Celsius. As you all know that if you increase the temperature, the rate of diffusion increases, right? So what you need to do, you have to choose an option in which the rate of diffusion will be slowest, right? Because you want a lighter blue color in water as compared to the first experiment. And the blue color in water will be lighter when less dye escapes the block and goes into water, right? So the temperature you must choose must be less than or equal to the temperature in the first experiment. So in options we have 20 and 30. So obviously when the temperature is 20, those will be the only options. A and D will be wrong. That's because the temperature is higher. If we take higher temperature, so the rate of diffusion will be faster and more dye will go in water from the block, and we will observe a darker blue color in the water. Fine. So the option could be B or D. C is wrong, sorry. A and C will be wrong because in these options the temperature is 30 degrees Celsius. B and D could be right. Okay. Now we have to decide on the basis of the size of each agar block and the number of blocks. How can we do that? Fine. Let's calculate the volume for the agar block that was used in first experiment. What is the volume? Volume is equals to length into breadth into height. So what is the volume of the agar block that was used in the first experiment? The volume is 1 centimeter into 1 centimeter into 1 centimeter. So the answer is 1 cm cube, right? Okay, let's calculate the volume for these blocks. If we calculate the volume 0.5 into 0.5 into 0.5, the answer will be 0.125. And how many blocks do we have? We have 2 blocks, that means the total volume is 0.25. And over here, if we find the volume 2 into 1 into 1, the volume will be 2 centimeter cube. So guys, always remember that if the volume of the block or blocks will be higher than the original experiment, this means that the block can lose more blue dye and the intensity of the blue color in the water will increase. So option D could not be the answer, it will be wrong, because in option D, we have one block that has a volume of 2 centimeter cube which can hold more blue dye. And when we place this block in the water, more dye can be lost and go into the water, right? And in this way, the blue color in the solution in the water will be darker than the original experiment. But if we talk about the option B, which is the right answer, there are 2 blocks with a total volume of 0.25 centimeter cube, which is less than the first experiment, that is 1 centimeter cube. So these blocks will hold less dye and less dye will go in the water when these blocks are put into the water. So the intensity of the blue color in the water will be lowest. The answer will be B. Okay, let's move on to MCQ number 18, which description is correct. A centromere holds 2 chromatids together until the end of the prophase and attaches to the microtubules of the spindle. The centromere holds the 2 chromatids together. This is true. A centromere always holds the 2 chromatids together, but not until the end of the prophase. Until the end of the metaphase. Why? Because when the anaphase begins, the centromere splits, and at this moment, the 2 chromatids separate. So until the end of the prophase is wrong in this statement. So option A is wrong. A chromatid is one of the 2 identical parts of a chromosome. This is true. Chromatid is one of the 2 identical parts of the chromosome. We have 2 chromatids. We have 2 identical chromatids in a chromosome. This is true. And is made up of proteins and 2 molecules of DNA. This is wrong. Chromatid is made up of proteins, but not 2 molecules of DNA. It is made up of 1 molecule of DNA. So option B is wrong as well. A chromosome is a structure with 2 identical parts. This is true. A chromosome is a structure with 2 identical parts or chromatids made up of DNA and proteins. This is true as well. Found in the nucleus of a prokaryotic cell. No, there is no nucleus in a prokaryotic cell, right? A telomere is a sequence of DNA nucleotides such as GGG, TAA repeated many times and found at the end of each chromatid. This is true. As you all know, that telomere is a sequence of non-coding DNA nucleotides such as GGG, TAA, which repeat many times. and they are found at the end of each chromatid or the end of the DNA. Right answer is D. Let's talk about MCQ number 19. During metaphase, a scientist stains the chromosomes of a diploid animal cell with the fluorescent dye to allow the telomeres to be observed. This cell has 26 chromosomes. How many telomeres will the scientist observe? Okay, so they are talking about metaphase. In metaphase, each chromosome has how many chromatids? Each chromosome has how many chromatids? Yes, guys, 2 chromatids. So each chromosome will have how many telomeres? 4 telomeres, right? Because at the end of the chromatid, we find a telomere. So during metaphase, each chromosome has 2 chromatids, and at the end of each chromatid, there is 1 telomere. So one chromosome contains how many telomeres? 4 telomeres. One chromosome contains 4 telomeres. Right? How many chromosomes do we have? 26 chromosomes. So 26 into 4 will be the telomeres found on 26 chromosomes. The answer will be 104. The answer is D. Let's move on to MCQ number 20. The photomicrograph shows cells in various stages of the life cycle. Let's identify these stages. So guys, one is metaphase. How do we know that this is metaphase? Because the chromosomes are in line. During metaphase, the chromosomes align at the equator of the spindle. So chromosomes are kind of in a line. This is metaphase. The phase 2 is the telophase because the chromatids have arrived to the opposite poles of the cell. So this is telophase, when there will be 2 nuclei in the cell. Right? Right now there are not 2 nuclei, but we can see that chromatids have reached opposite poles. So definitely this is the start of the telophase. If we talk about 3, we can observe the chromosomes, but these chromosomes are scattered. There is no nuclear envelope, right? It means this is prophase, late prophase. No nuclear envelope. We can see chromosomes which are scattered. And if we talk about 4 is anaphase. How do we know that 4 is anaphase? Because the sister chromatids, or we can say the chromatids, are pulled towards opposite poles with the V arrangement. Whenever you see the V arrangement, this is anaphase, in which chromatids are being pulled towards opposite poles. Which cells contain twice as many DNA molecules as a cell from the same organism that has just finished a complete mitotic cell cycle ending with cytokinesis? Guys, always remember that before DNA replication, always remember this point, that before S phase and after S phase, how the number of DNA changes. For example, if before S phase DNA is, for example, before S phase, if DNA is 46, then after S phase, the DNA will be 92. So DNA doubles after S phase, right? So this is the point that you should note. One more thing is that if we recall cell cycle, how many phases are there in the cell cycle? There is G1 phase, then there is S phase, then there is G2 phase, then there is M phase, and then there is cytokinesis. Guys, always remember that just after cytokinesis till S phase, what happens? The chromosomes only contain one DNA, right? Each chromosome consists of only one chromatid. There is only one DNA. But after S phase, what happens? The DNA doubles. And this DNA is doubled till cytokinesis. Each cell contains a double DNA if the cell is in G2 phase, M phase, or cytokinesis. After cytokinesis and before S phase, the cell contains half the DNA as compared to the G2 phase, M phase, and cytokinases. So they say that which cell contains twice as many DNA molecules as a cell from the same organism that has just finished a complete mitotic cell cycle ending with cytokinases. If a cell has completed the mitotic cell cycle and the cytokinases has also ended, it means that the cell is before S phase, while the phases metaphase, telophase, prophase, and anaphase are after S phase, in M phase. This means that all these phases 1, 2, 3, and 4, they all will have twice as much DNA as a cell which has just completed cytokinases. A cell which has just completed cytokinases has not entered S phase yet, which means that any cell which is after S phase, which is in G2 phase or M phase or in cytokinases, will have twice as much DNA as the cell that has just ended cytokinases and is before S phase. I hope you get this point. MCQ number 21. Uncontrolled cell division can result in the formation of a tumor. Which part of the cell cycle would take less time during the formation of a tumor? As I mentioned in my previous lectures, that when a tumor is formed, the cells divide in an uncontrolled manner and interface shortens. Right? A tumor cell divides very rapidly. And in that case, the interface shortens. However, the M phase and the cytokinases have same length of time, but G1 phase, S phase, and G2 phase, they shorten. It means interphase is shortened. Let's move on to MCQ number 22. A piece of DNA molecule contains 84 base pairs. This means that each DNA strand contains 84 nucleotides. Always remember that whenever they say that a piece of DNA molecule contains this amount of base pairs, this means that each strand contains 84 nucleotides. One strand contains 84 nucleotides, the second strand contains 84 nucleotides. This means that the base pairs will be 84 because the bases pair up. They form a pair and pair is a group of 2. The table shows the number of adenine and the cytosine bases in one or both of the strands in the piece of DNA molecule. How many guanine bases are present in the piece of DNA molecule? So we have to find the cumulative number of guanine bases in both the strands, strand 1 and strand 2. So how can we figure out this number? Let's write strand 1 and strand 2 and the number of and the bases. In the DNA we have adenine, thymine, cytosine, and guanine. So how many adenine do we have? In the strand 1 we have 28 adenine and in strand 2 we have 23 adenine. So for thymine we will have the opposite number. If there are 23 nucleotides or bases, if there are 23 adenine nucleotides in strand 2, this means that in strand 1 there will be 23 thymine because thymine pairs with adenine. So if S2 strand 2 has 23 adenine, strand 1 will have 23 thymine. If strand 1 has 28 adenine, so strand 2 will have 28 thymine, right? For cytosine, there are 15 cytosine in strand 1. So this means that there are 15 guanine in the strand 2, right? Let's figure out the guanines. The total number of bases in one strand is 84, and we will get the total number by adding the guanine base, which is right now unknown, that is X. If we add 28, 23, 15 and X, we can get 84. So simply what we have to do is we have to subtract 28, 23 and 15 from 84. The answer will be, if we subtract, the answer will be 84 minus 15 will be 69. (84 minus 15) will be 84 minus 10 will be 74. 74 minus 5 will be 69, and 69 minus 20 will be 49. 49 minus 3 will be 46, and then 26. 26 minus 8 will be 18. So over here we have 18. So 18 guanine in one strand and 15 guanine in the second strand, together they make up 33. So the answer is B. Let's discuss MCQ number 23. The messenger RNA sequence. The messenger RNA sequences of the 3 stop codons are shown. Which mutation in the template or the transcribed strand of a DNA sequence that codes for a polypeptide would cause translation to stop prematurely? So in this MCQ, they're asking that which mutation in the template or transcribed strand will result in the formation of a stop triplet that would lead to one of these stop codons in the messenger RNA, which would lead to the translation to stop prematurely. As we all know that these are the 3 stop codons, and one of these 3 are usually found at the end of the messenger RNA to stop translation. If these appear somewhere between in between of the messenger RNA, or somewhere in the center or before the end, so the translation will stop prematurely. For these stop codons to appear in a messenger RNA somewhere before the end, there should be a mutation that results in the formation of a stop triplet. A stop triplet is the code, it's a group of 3 bases on the template strand, which will be complementary to the stop codon. So the complementary triplet to the stop codon UAA will be A, TT. Complementary to UAG will be A, T, C, and complementary to UGA will be A, C, D. So these are the stop triplets, which if are present in the template strand somewhere in between, so they will lead to the formation of one of these stop codons in the messenger RNA, and translation will stop prematurely. So let's see which is the right option. Always remember this point, that before mutation the triplet was normal, it was not a stop triplet, and after mutation it will change into a stop triplet. So ATT to ATC, this option is wrong, because ATT is already a stop triplet. It's not a normal triplet. Same goes for ACT, it's a normal triplet. If we talk about ACC, it's a normal triplet, which if changed to ATT, that is the stop triplet, this mutation will lead to the formation of a stop triplet and the stop codon in the messenger RNA, which will stop the translation prematurely. The answer is C, right? Other options are wrong, because the initial triplet that they have mentioned is a stop triplet. Mutation will result in the formation of a stop triplet. The triplet is not initially a stop triplet, it's initially a normal triplet. After mutation, it will become the stop triplet. Let's move on to MCQ number 24. Which features of the companion cells are essential to their function? Okay. Number 1, they are connected to the sieve tube elements by plasmodesmata. Yes, this is the essential structural adaptation for the function of companion cells, because as we all know that to load sucrose into the sieve tube, the sucrose moves from the companion cell to the sieve tube elements by diffusion via the plasmodesmata. Number 2, they have thicker cell walls than a sieve tube element. This has no role in the function of the companion cell. The cell walls of sieve tube element, the cell walls of companion cells being thicker than the sieve tube element has no functional role as far as the companion cells are concerned. They contain a nucleus in mitochondria. Yes, this is an important structural adaptation which will help the companion cells to carry out their function. As we all know that companion cells carry out active transport of hydrogen ions, so they need mitochondria. And companion cells are making proteins. So for that they need a nucleus, because transcription occurs in the nucleus. And messenger RNA are formed which go and attach to the ribosomes. So companion cells do need a nucleus and a mitochondria to carry out their function, and the plasmodesmata connections between the sieve tube and companion cells are also essential, so that the sucrose can go from the companion cells to the sieve tube elements via plasmodesmata. So the answer is 1 and 3. B. Which statement about water movement in plants are correct? Water can pass through the cellulose cell walls. Yes, water can flow from cell wall to cell wall. Water cannot pass through the unlignified cell walls. This is true as well, because lignin is waterproof. It does not allow the water to pass through. Water can pass through the cell walls that contain suberin. This is wrong. Suberin is again waterproof. It cannot allow the water to pass. Always remember that water cannot pass through the lignin and the suberin. However, water can pass through the cellulose. So the answer is B, 1 and 2 only. 3 is wrong. MCQ number 26. The diameter of a tree trunk usually decreases slightly during the day. Guys, remember that the tree trunk diameter decreases because of transpiration. Always remember that transpiration is occurring during the day, and this transpiration generates a transpirational pull which pulls the water up the xylem vessels. As the water is pulled up the xylem vessels, a tension is created, cohesion tension is created in the xylem vessel, which causes the xylem vessel to narrow down, and the tree trunk diameter decreases. Always remember that higher the rate of transpiration, the more the diameter of the tree trunk decreases, right? The tree trunk diameter is inversely proportional to the rate of transpiration. So greater the transpiration, more will be the decrease in the tree trunk diameter. Which changes in the environmental factors during the day could cause the diameter to decrease even more? So to decrease the diameter even more, we need to increase the rate of transpiration. As we all know, if we increase the wind speeds, rate of transpiration increases. If we increase the temperature, rate of transpiration increases. But to increase the rate of transpiration, we need to decrease the humidity, right? So this is wrong. Increased wind speed and increased temperature is right. The answer is B. If you increase the wind speed and if you increase the temperature, rate of transpiration will increase and the tree trunk diameter will decrease even more. But if you increase the humidity, rate of transpiration will decrease, and this will increase the trunk diameter, not decrease. MCQ number 27. Which sequence of events could lead to mass flow in phloem sieve tube? Okay. So we need to put these points in the sequence in order for the mass flow to occur in the phloem sieve tube. Always remember this point, that for mass flow to occur in phloem sieve tube, there should be higher hydrostatic pressure in the source in the sieve tube and the lower hydrostatic pressure in the sieve tube in the sink. So let's write these points in the sequence, how there is a high hydrostatic pressure in the sieve tubes at the source and a low hydrostatic pressure in the sieve tube in the sink. Okay, so first of all, what happens is that the protons are actively pumped out of the companion cell into the cell wall. So fifth one is the most starting point. Okay, then what happens because of this? As the protons are pumped actively out of the companion cells, they go into the cell wall, and then these protons bring sucrose together with them. So protons and sucrose molecules move into the companion cell through a co-transport protein. So after 5 we have 2. Okay, once the sucrose comes into the companion cell, what happens? Sucrose then moves into the sieve tube element from the companion cell. So next point will be 1. Once the sucrose enters the sieve tube element, what happens? The water potential of the sieve tube elements decreases. And then what will happen? As the water potential of the sieve tube element decreases, water from the neighboring cells enter the sieve tube down the water potential gradient by osmosis. And then once the sieve tube element gains water, hydrostatic pressure in the sieve tube element is produced. So the last statement is 3. So the answer is 5, 2, 1, 4, 6. 3. The answer is D. MCQ number 28. What will happen as a result of blood pressure in the right ventricle becoming higher than the blood pressure in the right atrium? Okay, if we draw the right side of the heart, so guys, there is a valve between the right and the between the right atrium and the right ventricle. So what are they saying? That if the pressure in the right ventricle exceeds the pressure in the right atrium, so the blood will try to backflow into the right atrium. And to prevent the backflow, this valve, which is the tricuspid valve, will close. We can also call it atrioventricular valve on the right side. So whenever the pressure of the right ventricle exceeds the pressure of right atrium, the blood tries to backflow from the right ventricle to the right atrium from high hydrostatic pressure to low hydrostatic pressure. Always remember that blood flows from high pressure to low pressure. So if the blood pressure in the right ventricle exceeds the blood pressure in the right atrium, the blood will try to backflow from the right ventricle to the right atrium from high hydrostatic pressure to low hydrostatic pressure. This will cause the tricuspid valve to close to avoid the backflow. So the answer will be the right atrioventricular valve will close. That is the tricuspid valve. Answer is D. Okay, MCQ number 29. The graph shows pressure changes in the different parts of the heart during a mammalian cardiac cycle. W, X, Y, and Z indicate when a valve opens or closes. So as I taught you guys in the lectures, that these intersection points are very, very important. At these intersection points, the valves are either opening or they are closing, right? So always remember that this dotted line over here, this one, represents the atrium. So whenever the pressure of the atrium rises like this, it means the cardiac cycle has started. This is atrial systole or atrial contraction. So when the cardiac cycle starts, or when there is atrial systole, the intersection point right after that, you can label this as A. Then the next intersection point is B. Then the next is C. And the last one is D. For that, we have a mnemonic known as Baap Coco, which you can use for these 4 points. For point A, the mnemonic is BC, that is bicuspid valve closes at A. Bicuspid valve closes, or we can say atrioventricular valve closes, because bicuspid valve is also known as atrioventricular valve. Okay, for X, which is B point, you can say aortic valve opens. Aortic valve opens. Aortic valve is also known as semilunar valve. So semilunar valve opens. Okay, for C, we have AC. Aortic valve closes, or we can say semilunar valve closes. And at D, for D, we have BO, which means bicuspid valve opens, or we can say atrioventricular valve opens. For W, in this case, atrioventricular valve closes, right? For X, which is the point B, semilunar valve opens, or aortic valve opens. For Y, which is point C, aortic valve closes or semilunar valve closes. And for Z, which is point D, AV valve opens. The answer is B. This is the best mnemonic for you to memorize, in which you can deal with such MCQs. MCQ number 30. Which statements are correct? Compared with blood, tissue fluid has less protein and no red blood cells. This is true. If we compare the tissue fluid with the blood, tissue fluid does not have red blood cells, and tissue fluid has far less protein as compared to the blood, because the proteins do not leak out of the blood into the tissue fluid. So tissue fluid has less proteins, and these proteins are usually made by the cells in the tissue fluid. Okay. Lymph may contain lipids, carbon dioxide, and phagocytes. This is true. Lymph inside the lymph vessels has phagocytes in it, and also has carbon dioxide and lipids. Tissue fluid contains glucose, amino acids, urea, and carbon dioxide. This is true. Tissue fluid is a medium between the blood and the cells of the body, and tissue fluid also contains nutrients and waste products. So tissue fluid can contain glucose, amino acids, urea, and carbon dioxide. The answer is A, 1, 2, and 3. These all statements are correct. Okay, MCQ number 31. Which statement about the formation of hemoglobinic acid are correct? it can only occur with the dissociation of oxygen from hemoglobin. This is true. For hemoglobinic acid to form, oxygen must dissociate from the hemoglobin. Right now we have oxyhemoglobin, hemoglobin to which oxygen is attached, and this is hydrogen ion. So for hemoglobinic acid, that is HHB, for this acid to form, oxygen must leave the hemoglobin. So first statement is true. It removes excess hydrogen ions, preventing blood from becoming too acidic. This is true. Excess hydrogen ions are picked up by the hemoglobin to form hemoglobinic acid, which prevents the blood from becoming too acidic. Statement 2 is also right. Statement number 3, it is linked to the action of carbonic anhydrase. This is true as well. Why? Because to make hydrogen ions, we need carbonic anhydrase. As you all know, carbon dioxide combines with water to form carbonic acid, and this reaction is catalyzed by carbonic anhydrase, and then carbonic acid dissociates into hydrogen ions and hydrogen carbonate ions. So for the formation of hydrogen ions, you need carbonic anhydrase, and for the formation of hemoglobinic acid, you need hydrogen ions. So we can say for the formation of hemoglobinic acid, we need hydrogen ions, and for the formation of hydrogen ions, we need carbonic anhydrase. So this reaction is linked to the carbonic anhydrase. The answer is A. All the statements are correct. MCQ number 32. What explains how the maximum volume of oxygen is taken up as the blood passes through the capillaries of the lungs? So as the blood passes through the capillary of the lungs, which statement explains that maximum volume of oxygen is taken up by the blood? Each hemoglobin molecule can temporarily bind to 4 oxygen atoms. This is wrong, because each hemoglobin molecule can temporarily bind to 4 oxygen molecules, guys, not atoms. This statement is wrong. The dissociation of carbon dioxide from the carboxyhemoglobin. Carboxyhemoglobin, are you sure? Carboxyhemoglobin is a hemoglobin which contains carbon monoxide, not carbon dioxide. This statement is again wrong, right? If carbon dioxide is attached to the hemoglobin, carbon dioxide is attached at the amine groups of the end terminals of polypeptide chains, forming carbaminohemoglobin, not carboxyhemoglobin. The binding of the first oxygen molecule to hemoglobin decreases the affinity. No. Whenever the first oxygen molecule binds to hemoglobin, the shape of the hemoglobin molecule changes, and this increases the affinity of hemoglobin for oxygen. This is known as cooperative binding. Whenever first oxygen molecule binds, the shape of the hemoglobin molecule changes, the affinity of hemoglobin for oxygen increases, and the next oxygen molecule can bind more easily to the hemoglobin molecule. This is called cooperative binding. Obviously, all the other options are wrong. This means the answer is D. Oxyhemoglobin formation increases the ability of the red blood cells to transport oxygen. This is very true, because once one oxygen molecule binds to hemoglobin, an oxyhemoglobin forms. So the shape of the hemoglobin molecule changes. Hemoglobin now can bind more oxygen molecules more easily, and this increases the ability of the red blood cells to carry and transport oxygen. The answer is D. MCQ number 33. The image shown in the photomicrograph, the image shown is a photomicrograph. That is the picture taken from the light microscope of a transverse section of the part of the gas exchange system. So guys, this is the part of the gas exchange system. What we can see is this is the ciliated epithelium. This is the ciliated epithelium, which contains the ciliated cells and the goblet cells, right? We can see this is the goblet cell. Okay. Then there's a connective tissue beneath the ciliated epithelium. This connective tissue contains elastic fibers and smooth muscle. Okay. What else? Then we have this tissue which contains glands. How do you recognize the glands? Whenever you find many nuclei, when you find a lot of nuclei, this means that these are glands, because glandular cells are enormous in number. So whenever you see a lot of nuclei, this means that these are the glands, and the glands that are found in the gas exchange system are the mucous glands. And this is the cartilage. As we can see, whenever you see dispersed cells, Whenever there are a lot of gaps between the cells, this is the cartilage. Okay. What describes the image? A thin layer of ciliated epithelial cells on top of a layer containing cartilage supported by elastic fibers. No. A thin layer of ciliated epithelial cells on top of a layer containing cartilage. No, ciliated cells are not on top of cartilage. On top of cartilage, we have the mucous glands, right? Just right on top of the cartilage. We have the mucous glands, then we have the elastic fibers. A very thin epithelial lining with walls containing elastic fibers surrounded by many blood vessels. We cannot see any blood vessels over here. We cannot appreciate the blood vessels in this diagram. So this statement is wrong as well. There is a very thin epithelial lining fine with walls containing elastic fibers. This is true, but surrounded by many blood vessels, we cannot see any blood vessel over here. An inner layer of ciliated epithelial cells and goblet cells on top of elastic fibers. This is true, supported by an outer layer consisting of cartilage. This is the perfect statement. C is the answer. An inner layer of ciliated epithelial and goblet cells on top of loose tissue with mucous glands. No, it is on top of the elastic fibers, not on top of the loose tissue containing mucous glands. Mucous glands are below the elastic fibers, supported by a continuous ring of cartilage. We cannot appreciate if there is a continuous ring of cartilage. This might be a bronchus with irregular blocks of cartilage, right? We cannot appreciate on this diagram that there is a continuous ring of cartilage. So D could not be the answer. And also, the ciliated epithelial layer is above the elastic fibers, not above the loose tissue containing mucous glands. MCQ number 34. Which row shows the tissues that are present in the wall of the trachea and the wall of the bronchus? So in the wall of the trachea and in the wall of the bronchus, we have the smooth muscles. We don't have squamous epithelium because squamous epithelium is found in the alveoli and the alveolar ducts. In the wall of the trachea and the bronchi, we have the columnar epithelium. Goblet cells are present in bronchi and trachea. The answer is C. MCQ number 35. When a person suffers an asthma attack, the tubes of the gas exchange system narrow and extra mucus is produced. Okay. So the information says whenever a person suffers from an asthma attack, what happens that the tubes of the gas exchange system, they become narrower, which means that smooth muscles are contracting. Whenever the smooth muscles are contracting, there is bronchoconstriction, and due to bronchoconstriction, the tubes of the gas exchange system narrow down. And when the extra mucus is produced, it means that goblet cells are working more and the mucous glands are working more. Which changes occur during an asthma attack? Activity of ciliated epithelium increases. This is true because ciliated epithelium contains goblet cells. Goblet cells are more active, they're producing more mucus. Similarly, obviously, if more mucus is produced, so more cilia are working. So the activity of ciliated epithelium increases. This is true. Endocytosis in goblet cells increases. This is wrong. Why? Exocytosis occurs in the goblet cells, not endocytosis. When extra mucus is produced, it means the vesicles are fusing with the membrane and contents are released out of the cell by exocytosis. Smooth muscles are more active. This is true. So the answer is 1 and 3 only. The answer is C. Which flow diagram correctly describes the effect of tar entering the lungs? Whenever the tar enters the cells of the lungs, tar contains carcinogens that come into contact with the DNA. This is true. And this will lead to mutations. Very good. And eventually mutations in the proto-oncogenes will result in the formation of oncogenes that will result in uncontrolled cell division. The answer is A. Okay, goblet cells secrete more mucus. Guys, in the lungs, if we talk about specifically about the lungs, so we are talking about alveoli. Goblet cells are present in the trachea and bronchi, not inside the lungs. Okay. The other options are wrong. The answer is A for 36. 37. Which control measures would reduce the transmission of TB? Boiling water before drinking it. Guys, TB is transmitted by respiratory droplets. It's not transmitted via water. So even if you boil the water before drinking it, it will not reduce the transmission of TB. Antibiotics, yes, antibiotics will reduce the transmission of TB because TB is caused by bacteria and antibiotics kill bacteria. Vaccination against TB will also help to reduce the transmission of TB. The answer is D. Comorbidity is where an individual has two or more diseases or medical conditions at the same time. Okay, so they are defining comorbidity. It's a condition when the individual has two or more diseases at the same time. Some medical conditions are particularly likely to result in comorbidity. In one example, initial infection with one pathogen can increase the risk of developing a second disease by a factor of 15 or more. The second disease is then the major cause of death for these people. I hope you get this information. They're saying that comorbidity, for example, one disease can increase the likelihood of getting another disease. One pathogen can cause the development of another disease by a factor of 15 or more. So if a person has an infectious disease, there's a likelihood of getting another infectious disease as well. For example, if you have a throat infection caused by a virus, you can get a superimposed bacterial infection in your throat. Which description could fit this example? Cholera infection followed by development of malaria. This never happens. The infection, if you get infected with malaria, you don't get cholera. Cholera infection followed by development of AIDS. No. HIV infection followed by development of TB. This is true because once you get an HIV infection, once you get AIDS, you can easily develop TB, right? But for cholera infection, after you get the cholera infection, you will never get AIDS. However, if you get the AIDS first, then you can develop cholera infection, because they say cholera infection followed by development of AIDS, which means cholera infection will occur first and AIDS will develop later. No. If you get cholera infection, there are no chances that you get AIDS. But if you get AIDS, you might develop cholera, which is not the option over here. But whenever you get an HIV infection, we studied this in our lectures, that whenever we get HIV infection, whenever a person develops AIDS, the person develops TB afterwards very easily. MCQ number 39. Which conditions are infectious and result in high white blood cell count? So there are two words over here which you need to keep in your mind. The disease should be infectious and it should result in a high white blood cell count. Leukemia is a blood cancer. It's not an infectious disease. It's not caused by pathogen. Leukemia results in a high white blood cell count. This is true. Leukemia is a blood cancer and this results in the high white blood cell count, but it's not infectious. It's not an infectious disease. It's not caused by a pathogen. It's a cancer. If we talk about myasthenia gravis is an autoimmune disease. It results in the high white blood cell count, but it is not infectious. Myasthenia gravis is now, it's not in your syllabus anymore, but you should know it's an autoimmune disease, and autoimmune diseases increase the white blood cell count, but they are not infectious. If we talk about TB, yes, TB is infectious, and also it also results in the high white blood cell count. Obviously, there's an immune response when the mycobacteria enter the body. So white blood cell count increases, and this disease is also infectious. It's caused by a pathogen, Mycobacterium tuberculosis and Mycobacterium bovis. Okay, so the answer is D, 3 only. Which cell type causes the secondary immune response to be much faster than the primary immune response? Always remember that secondary immune responses are way too fast than the primary immune responses because of the memory cells. The answer is B. So this concludes our paper, that is October-November 2021 AS level biology 9700. Thank you so much for watching.